DNA,
Recording in progress.
and that resulted in no transformed cells, right? And today, of course, it makes sense because, if the rough cells had no intact gene to pick up and then transcribe and translate it so that they could synthesize a capsule, of course, they're not going to be transformed. So, that didn't work out, and that obviously made sense. And then finally, people were interested in learning whether RNA, okay, that might have contaminated this DNA extract could be the genetic material. So they added RNAs, and adding the RNAs had no impact on the transformed cells. You still observe transformed cells. And similarly, DNA can be in complex with lots of proteins. So people wanted to know perhaps it's the contaminating proteins that are responsible for this, transformation. So proteases were added, and the result was that there were, still transformed cells. So taken together, you can see that absence of proteins has no impact on transformation. Absence of RNA has no impact on transformation, but absence of DNA does have an impact on transformation. So it means that DNA is that transforming principle that the rough cells were picking up and becoming a different type of a cell in the process. So in another 10 years, okay, people are still sort of debating how can DNA be the genetic material, being that it's only comprised of 4 nucleotides. So Hershey and Chase experiment, the famous experiment that you guys have studied again in introductory bio, sort of confirmed this idea of DNA being the genetic material using viruses as a model organism. Viruses are sort of very unique in that they're very simplistic, right? They're just two things that they're made up of. They are made up of a outer protein shell that surrounds the genetic material. So they were lucky in that they chose a E. coli phage that has DNA as the genetic material. And so they labeled their viruses in two ways. Either they labeled the coat, okay, the outer shell, that is only made up of protein. They either labeled it with the radioactive sulfur because sulfur is only found within proteins, and they knew about the life cycle of this bacteriophage. They know that the protein coat remains outside of these bacterial cells. The genetic materials is injected, and so they allowed this initial injection and the lytic process to begin. But once the DNA has been injected, they sort of whirl the culture with bacteria that have already been injected. There's to kind of give it a real whirl in a blender, and that caused all of these virus shells to fall off. And so the DNA that they had injected, which would eventually cause the host cell to produce new viruses, has already been injected. And so beyond this, the shell isn't there, but the bacterial cell is able to still undergo the lytic cycle, produce new virions. Okay, so that is obviously going to happen under the directions of the genetic material of the virus, right? So whatever was the genetic material has already been added or injected into the host cell. So the second experiment was now this time radioactively labeling DNA. And DNA is unique in that it has phosphate groups that proteins don't have. And so they were able to demonstrate that now, when they repeated the experiment exactly the same way, the radioactively labeled DNA was found to be within the bacterial cells. Okay. And, these cells would go on to then undergo lytic cycle and produce new virions. So, Hershey and Chase were finally able to put this debate to rest, and they were able to demonstrate that it was the radioactively labeled DNA that was injected inside the cell, not radioactively labeled protein, and it was the DNA that was controlling the host cell to produce new virions. And so, it took a while for us to kind of get to here, but this was the right conclusion. Haley, you have a question.
Yes, Professor, I do. Sorry. I read the book and the experiment. I still not fully understand this experiment. So they label the protein to know if the bacteria will eventually do progenies of the bacteria. I didn't fully understand the experiment.
I'm sorry, you're breaking up a little bit, so I couldn't fully hear your question at
Okay,
the
yeah.
end.
Can you hear me now?
Yeah, I can hear you.
Yeah, what I'm saying is, I did not fully understand this experiment. I'm not sure exactly what they're trying to demonstrate. Is it why they're trying to say if the DNA is the one that
Mhm.
caused the virus or the protein?
Right. So what there's just two things, right? So if you could somehow have a way of seeing them, right? And we know already that the viruses are going to basically introduce their genetic material into the bacterial cell. People knew a lot more about viruses in like the 1920s, 1910s. So people had already sort of deciphered the life cycle of viruses. So they knew much more about viruses than they did about the cells, because cells were much more complex. They have many more enzymes, they have a lot going on compared to viruses. So they're just two components. They're made up of either protein or they have this genetic material DNA. If we could have a way to see both the protein and DNA, which they do by radioactively labeling the protein with S35 and radioactively labeling the DNA with P32. And if they could demonstrate which of these two radioactive compounds enters the cell, then they know that that compound is the one that is responsible for overtaking the host machinery and causing it to produce more virions, right? So that's got to be the genetic material. So doing this experiment, they were able to demonstrate that it was P32, which corresponds to DNA that had entered the cell and then resulted in the host cell producing more virions, right? The protein remained outside of the cell. You shook it off. It didn't have to be even near the host cell in order for it to produce any more, virions. So it's in a nutshell, that's what the whole goal was, and that's what it accomplished. Is that a little bit clearer?
Yeah. So at the end, so when they checked the bacteriophages, they did not find any protein, but however, they do find DNA inside of the bacteriophages.
So here is are you confused here?
Yes, that's why this is exactly what I'm confused between these two points at the end.
Okay, all right. So here I don't, so I think they're just showing you the blender treatment. They're not showing you what you're going to have at the end. So unless these bacteria were growing in a radioactively labeled um, um, you know, um, culture where they had radioactively labeled amino acids or nucleotides, the virions that are going to be produced are not going to be labeled. Okay, so this is just showing you at the end of the blender treatment. So you give it a whirl, the protein shell falls off, and the genetic material remains within the cell. Okay, so in this case, the genetic material was not labeled, so you're not going to see it inside the cell, right? In this case, the genetic material was labeled, so you're going to see it inside the cell. Um, I think don't consider these viruses as the newly synthesized virions. These are just ghosts of the original phages that had infected the host cell.
I see. Okay, I see. Thank you so much.
Okay, sure, no problem. Okay, so now going back to subunits of DNA and RNA, of course, you guys are aware of it. Again, should be a reminder. The nucleotides generally are comprised of a nitrogenous base, which come in two forms. You can have purines and pyrimidines. Purine bases have two rings, as shown here. All of these are purine rings. Pyrimidines are comprised of a single ring. So purines are your adenine and guanine, and your pyrimidines are your cytosine, your uracil, and thymine. Okay, and these nitrogenous bases are attached to the one prime or the carbon position of the ribose sugar. In DNA, it's the deoxyribose sugar. What that means is that it's missing a hydroxyl at the 2' position. So you're going to have two hydrogens here. In RNA, it's the ribose sugar. So you're going to have two hydroxyls at both at the 2' and at the 3' position. This 2' position is really important to differentiate between, I guess, the RNA and the DNA nucleotides, but it's really the 3' hydroxyl that is really important the polynucleotide formation reactions, as we're going to see, or polymerization reactions for DNA and RNA. So, when we talked about last time drugs, and we talked about certain drugs that were used to inhibit HIV synthesis, and we talked about how there could be nucleotide analogs. These analogs are often missing these hydroxyl groups. So once they're inserted, they can't be used to add additional nucleotides on. All right. So this is kind of like, that unit on a building block that is used to fit further building blocks on top of it. If it's malformed, or if it kind of doesn't fit the plan, then you can't add any more building blocks. So this 3' hydroxyl position is really, really important. And then you have the 5' position, and that 5' hydroxyl is attached to phosphate groups, right? So generally we have the 3 phosphates. So we can have, ATP or GTP or TTP and so on. Here you're seeing the adenosine, and you're seeing 2-deoxyadenosine monophosphate. It should be, triphosphate if it's the nucleotide that is going to serve as a subunit for polymerization. This is something that will exist as part of the polymer as just one phosphate, monophosphate. Adenosine, and interestingly, I find it really interesting that it's actually found as a neurotransmitter within our brains. It's found as a hormone within our blood as well. So it acts as I believe, as a depressant sort of a neurotransmitter within your brain, and acts to slow down your heart. And it's often used as a drug to treat arrhythmias like tachycardias and so on. So, I know it doesn't have anything to do with the bacteria, but we have the same nucleotides, and it's kind of interesting to think about how you can have these nucleotides that are subunits for DNA, such an integral part of your genetic material, yet your body has the ability to also use them as neurotransmitters and hormones. Really, really interesting. And we exploit that, right? We know it. We can synthesize adenosine, and we can use it as a drug to treat arrhythmias in individuals. But anyway, so you have this basic structure. So when we talk about 5' to 3' growth of our polymers, we're basically referring to this sort of polarity of the molecule, that on one end of your DNA or RNA, you're going to have the 5' phosphate group, and on the 3' end, you're going to have a hydroxyl group. And anytime we talk about growth of DNA, you can add to that 3' hydroxyl. You cannot add to the phosphate group. All right. So this end doesn't grow. This end always grows. And this is an important understanding to have at the back of your mind, especially when we're talking about, replication and transcription. Okay. So 5' to 3' end, when you read about it, just bear in mind that these are the two sort of groups that we're referring to, and the 3' hydroxyl group is going to generally be the end that grows. Now, in terms of DNA structure, again, something that you were aware of, DNA is comprised of 2 polymers of nucleotides, and they run in anti-parallel direction. So on the strand on the left-hand side, I'm seeing a phosphate group here. So that's a 5' end. And if I continue to kind of go down the strand all the way to the other end, I'm seeing a hydroxyl group. So this is a 3' end. So if I were going to add additional nucleotides, I could only add it to this hydroxyl and then kind of continue to grow the chain towards the bottom of the slide. Okay, so this is one way that the nucleotide is running. The other nucleotide polymer is running in the opposite direction. You have the 5' end at the bottom and 3' end at the top. So this will grow in the opposite direction towards the top of the slide if we were to continue to use this hydroxyl to grow additional or add additional nucleotides to it. So it's anti-parallel, and it's important because when we talk about replication, the cells are going to be have to be mindful of that, right? That they will be growing the two strands of DNA in opposite directions, right? Yet replication fork is going to move in only one direction. So they're going to have to kind of bubble out one of these strands and use that as a template while, synthesizing the other one without having to make that bubble. So we'll talk about that in a little bit. So the other thing that you should probably think about is that, there are different forms of DNA. There is the A form and the B form and the Z form. This is the B form, and generally it's defined by having 10.5 base pairs per turn, and it has a particular height of, I believe, 3.4 angstroms. And so what this is telling you is that it has this uniform diameter. Again, that uniform diameter is kept uniform because you have this base pairing that is between the complementary bases, right? So you always have a purine against a pyrimidine, and specifically a G against a C and a A against a T, right? And oftentimes when DNA polymerases are adding these nucleotides against, a template, they're going to look for the number of hydrogen bonds that exists between the bases, right? So when DNA polymerase adds a C against a G, it looks for how many, hydrogen bonds there are. If it finds that there are accurate number of hydrogen bonds there, it considers this to be a correct insertion. If there is an inaccurate number of hydrogen bonds, it removes the most recently added base and puts in a brand new one. Okay, so it's important that the polymerase ensure this uniform diameter. Okay, and the other thing that you should keep in mind is that there are two types of grooves that form. There is the major groove that generally is going to help the um, activators for transcription or repressors for transcription kind of sit in on certain sequences within the DNA. And then there is the minor groove that doesn't generally serve that purpose. Okay, so when we talk about repressors binding, next time when we talk about transcription regulation or activators binding to promote transcription, we'll refer to this major groove. And major groove is this area that sort of is more pronounced, this groove that is more pronounced versus minor that it isn't as pronounced. So this is generally the form that is found as the chromosomal piece within bacteria, and it's generally negatively it's not, it could be negatively supercoiled, it could be positive in certain places. It depends on how actively transcribed a particular region is, right? So the bacterial chromosomes, just like eukaryotic chromosomes, is not going to be just sort of this uniformly supercoiled. It's got to have some regions that are tight versus other regions that are kind of loosely supercoiled. So when we talk about negative supercoiling and positive supercoiling, what does that mean? Negative supercoiling means that there are going to be fewer base pairs per turn. Okay, so it's going to be a tighter coil. Okay, if you consider this a spring, you can consider a wider spring and a narrower spring. Like a narrower spring is going to have, a fewer, thread, for example, or fewer metal that makes up that that individual turn. But you consider a loose supercoil, it's going to have many more base pairs per turn. And so you can have positive supercoiling. That means that you have a loose DNA strand that is probably actively being transcribed. And you can have negative supercoil where the sequence is much more tightly coiled. So there are fewer base pairs per turn. And it's probably not being actively transcribed at the moment. So you're going to see this phenomenon in all types of cells. And then you can have other forms of DNA, the A form and the Z form, which A form, I believe, has 11 base pairs per turn, but it's actually wider in diameter than the B form, and Z form is actually even narrower in diameter, but it has even more, base pairs per turn. Generally, B form is the form that we see in being the predominant form within cells. The first thing we'll talk about is DNA replication, and the process is very similar to that you have seen in eukaryotic cell in what we refer to as the semi-conservative replication. In semi-conservative replication, you're going to have the parental DNA strand that is going to separate into individual polynucleotides, and the exposed bases will serve as a template to synthesize individual daughter strands. All right, so you're going to have this newly synthesized DNA molecule that is half the original strand, the parental strand, and half the daughter strand. So the idea here is that the semi-conservative replication is going to guarantee that the genetic material would not vary from the parent to the daughter cells. Okay, and we're going to see that bacteria have the ability to go back, and in case if there were wrong bases incorporated, they have the ability to go back, look at the daughter strand, and replace those mismatched bases, as we're going to see next time. So they must have a way of then distinguishing between a parental strand and a daughter strand, or the new strand that was just synthesized. But this sequence of bases is extremely important because that is what the genetic code is. You have to maintain those sequence of bases from one cell to the next if you don't want the cells to change too much, and that's what we see um, in bacterial, division. We know that there is no process of meiosis where there's no sort of genetic reshuffling, and so the genetic code is really well maintained, and so it's the sequence of bases that need to be conserved, and by using one of the parental strands as a template, that process is ensured. Now, cells will make an error every now and then. Okay, if we consider E. coli, it can make an error for one error for every million genes that it copies, which is very, very small. So it means that bacteria then have this sort of proofreading mechanism that's going on as they are copying the genetic material. So in terms of eukaryotic cells, you remember that you have multiple chromosomes. Considering human, say, cells, we have 46 chromosomes, and each one can have millions to billions of base pairs of DNA. And so there are multiple regions where replication can start that we can refer to as origin of replication. And often these regions tend to be rich in adenines and thymines, or A's and T's. And the reason is that these A's and T's will be held together via two hydrogen bonds, and it's a little bit easier to break two hydrogen bonds. They're going to require less energy versus breaking bonds between G's and C's. So you're going to have these origin of replication within bacterial chromosomes as well, but bacterial chromosomes have a single origin of replication. Okay, versus multiple per chromosome in eukaryotic cells. And so this origin of replication has to be recognized by the replication machinery when the cell commits to cell division. And the replication fork kind of There's two of them. Replication fork is just the area where actually copying of the DNA is occurring. There's going to be two replication forks that will kind of move away from origin of replication in opposite directions. Okay, and they will continue to kind of copy, you know, moving away from origin of replication all the way to the other end opposite the origin of replication, that is referred to as the termination sequence of the terminator, written here as TER. Okay, so origin of replication, without the origin of replication, you're not going to be able to copy the genetic material, right? You might have it, you might have all the enzymes needed to copy it, but if you don't have that initial mechanism of recognizing where to begin this process, replication will not happen. Similarly, termination is required. The sequence is required for the forks to kind of recognize this is the end of this process and then, stop and fall off. But fortunately for the terminator sequence, you can have a few mutations and the replication can still stop because the two forks would eventually run into one another, right? Each one is really copying half of the chromosomes. The two will run into one another at the terminator sequence and kind of, fall off. And so you would have, of course, a fully copied genetic material. And so you have this entire chromosome being copied as a single unit. So we refer to it as a replicon. In eukaryotic cells, that's not the case. You're going to have multiple replicons per chromosome. Now, one of the things to keep in mind when DNA is being copied, whether it's eukaryotic cells or prokaryotic cells, is that you have this coil that you are unraveling, right? You started off with this closed circle, which is sort of, this two polynucleotides that are wrapped around one another, and you're going to begin to unravel that at the origin of replication. You're going to begin to use the exposed parental nucleotides to add new ones into the daughter strand. And what this does is that it produces, this supercoiling of the region ahead of the replication fork. And it's extremely important for bacterial cells to release some of this tension in the DNA that lies ahead of the replication fork if you want the replication fork to pass through. All right. So it's going to have, bacteria are going to have to rely on enzymes that we refer to as topoisomerases that come in, cleave just one of these two DNA strands, maybe just the blue one, and it will just kind of quickly, unravel, releases a lot of the tension. They remain kind of attached on both ends that were cut. And then once that tension is gone and you don't have that rapid unraveling going on, they will help to reseal that cut as well. So topoisomerases are going to make a cut, allow the supercoiling to be sort of relaxed, and then they will reseal that cut so that there's no physical open space within the template and the replication fork can easily pass through. All right, so topoisomerases are extremely important. Gyrase is going to be an example of a topoisomerase whose job is to continuously lie ahead of the replication fork and continue to sort of release this supercoiling, this tension that lies in the template ahead. Okay, and we talked about some drugs that happen to target these types of enzymes, the topoisomerases and gyrases and things like that. And in absence of those enzymes, the replication cannot be completed because you can't simply go through, the replication force cannot simply pass through that overly supercoiled region and then fully complete the replication process. So, cell will just kind of stop at this step where a portion of the DNA has been replicated, but the replication cannot be completed. If you can't complete the replication, then you don't have enough genetic material to pass on to your daughter cell. So you remain stuck being yourself, right? And so eventually either the cell dies or the immune system cells would recognize it as a threat and destroy it. Okay. But so, topoisomerases are extremely important. So here again, just kind of showing you the positively supercoiled, meaning that it's going to have, fewer base pairs. I'm sorry, more base pairs um, per turn versus negatively supercoiled, which is, tighter coils, so fewer base pairs per turn, and relaxed is what's required for the replication machinery to really pass through, what's required for transcription machinery to pass through. And if you have relaxed one relaxed area, it definitely means that there are other areas on the genetic material that are negatively supercoiled. Okay, so topoisomerases are extremely important to release some of that tension. Now we're going to start talking about replication first. And so there are many different proteins that are involved in carrying out the replication. So replication is simply just copying of the parental strand into the two daughter strands. And so we're going to look at all of these proteins as we talk about the process. This table kind of lists them out for you in case you need to go back and you need to look at what exactly they do. So replication machinery is comprised of a DNA polymerase. Okay, polymerase basically just takes subunits and links them together into a polymer. So you're going to have DNA polymerase that's going to make a polymer of deoxyribonucleotides, right? And you can have RNA polymerase that can link together RNA nucleotides to make, ribonucleotide polymer. And so DNA polymerase reaction is that it can take any of the DNA nucleotides in presence of a template. It is able to put the two together. And keep in mind that the energy for this process comes from the breakdown of the pyrophosphate bonds. So you'd remember when you break down ATP, you just sort of remove the terminal phosphate group. So in this case, we're removing two phosphates. So this is equivalent of hydrolyzing two ATP molecules. And so these are all those energy carrying molecules within the cell that we're linking together essentially, right? So you're taking a bunch of ATP, right? And you're linking them together to make this polynucleotide that we refer to as, say DNA or RNA. So no additional energy would be required. The substrate itself will provide those high energy phosphate bonds, and you're not breaking one, rather you're hydrolyzing two phosphate bonds to release all that energy that is required by this DNA polymerase. So E. coli has several DNA polymerases. We're going to talk about two of them today. Polymerase 3 is the one that is mainly responsible for just copying the template into the daughter strands. That is what it does, okay? And when we look at this DNA polymerase, it doesn't sort of act alone. You have several of these polymerases that work together. So these yellow proteins that you're seeing, these are your DNA polymerase 3, okay? And they act together with a beta clamp that helps to kind of leave this enzyme attached to the template so that it doesn't fall off as it's making a copy of the DNA. And then you have this tau complex whose kind of job is to essentially hold the three of them together, right? There is evidence there might be more in some cases. There doesn't have to always be three. You can have more than three core enzymes as well. But so the tau complex, its job is to kind of hold them together. And also it helps to allow this beta clamp to latch onto the template and then it remains attached. So you at least have sort of these three of these core enzymes that are present in one giant, holoenzyme complex. And not only is that this core enzyme is able to do this polymerization reaction, it is also able to do the reverse, okay, which is the proofreading activity. It generally grows DNA from 5' to 3', meaning it can only add additional nucleotides to an existing 3' hydroxyl group, but it also has the ability to step back and look and check if it was the correct nucleotide or not, and remove the nucleotide by stepping back, meaning it's going to move from 3' to 5' direction. Okay, so it does both things, and so its proofreading activity generally resides in the domain referred to as the epsilon. Okay, epsilon domain has the proofreading activity. So if you were to mutate just the epsilon domain, the polymerization activity would remain, so that the polymerase would continue to add nucleotides, but it would not be able to go back and check and remove the wrong nucleotides and insert new ones. Okay, so it's important that there are different regions or domains within this polymerase that are responsible for different functions. Okay, so there can be strains that mutate very rapidly, and it could be due to changes in their polymerases that prevent them from making these repairs or proofreading as they are making copies of their genetic material. Okay, so again, just kind of reinforcing that idea that it's the 3' hydroxyl that is required to make an attack on the phosphate group of the 5' carbon of the incoming nucleotide that would eventually result in synthesis of the phosphodiester bond. Okay, so this is your phosphodiester bond that is going to link all the nucleotides and the subsequent nucleotides together as well. And the release of these two phosphate groups is equivalent to spending 2 ATPs at each of these steps. And so the next nucleotide would now be added to the next 3' hydroxyl that you see coming up, right? So you're growing this chain from this 5' end to 3' end, then you will, put in a new nucleotide, and that 3' end would remain. So you're growing this 5' to 3', but when you were proofreading it, the DNA polymerase has the ability to move back, kind of slide backwards and go from 3' to 5' end to perhaps remove this nucleotide if it was inaccurate and replace it with an accurate nucleotides. Okay, questions? Okay. All right, so more replication protein. So we know what that polymerization reaction is. We know that DNA polymerase 3 is responsible for generating that phosphodiester bond, right? It relies on the template strand in order to see what nucleotide to insert next. If it sees a G, it's going to insert C opposite it. If it sees an A, it's going to insert a T opposite it, right? So we know that complementary base pair ruling already. So in terms of, how does this process start and what are some of the other helper proteins that we saw on the table, what it is that they are doing. So DNA A is responsible for binding to that origin of replication. ORI-C is the origin of replication for E. coli. You will see this often, especially if you've done any type of plasmid transformation and you see ORI-C. This defines the origin of replication for plasmid in E. coli. So if you were to introduce this plasmid in E. coli, this same replication machinery would recognize this origin of replication and make copies of the plasmid as well. All right, so there are some origins of replication that allow copying of this plasmid only once every time the genome is copied. And then there are other slightly mutated ones that allow multiple rounds of replication to occur, for the plasmids, even if the genome isn't copied, even if the cell isn't undergoing cell division. So you can have multiple copies of a plasmid present within cells, which then they can share with other bacteria as well. But as I mentioned earlier, the origin of replication is going to be often rich in A's and T's. It does not mean that you're not going to have G's and C's. It just means that you're going to have a lot of those around so that it makes melting that area possible. So DNA A is often just sort of that flag that sits on this origin of replication and calls the rest of the members of the replication machinery to that area that we're ready for this process to begin. So once the origin of replication is recognized, then you have an enzyme called helicase or DNA B that comes in and begins to break down the hydrogen bonds in that region, right? You have to initially expose the nitrogenous bases by keeping them apart. You need to melt the hydrogen bonds that are holding the A's and T's within that region. So that's DNA B or helicase's job to come in and do that. So DNA B tends to remain associated at each of the replication forks, right? Because you're not going to melt the entire template all at once. You're going to melt it little pieces at a time. And the reason is that as soon as the helicase has gone through, it's going the nitrogenous bases can reattach. They can reform the hydrogen bonds that were just broken. Okay, so it's important that DNA B kind of remains at the forefront of this replication fork and kind of continue to melt the DNA a little bit at a time. Now, once it melts it, it exposes these regions of single-stranded DNA that either need to be, copied immediately. So that's what we're going to see in the leading strand synthesis, that as soon as the helicase passes through, the DNA polymerase 3 is able to replicate one strand of the DNA immediately in what we call the leading strand synthesis, so that you have the DNA polymerase that is kind of just trailing the helicase on that strand. So it's kind of keeping up with as the DNA is being unraveled. Now, on the opposite strand, you have regions of DNA, single-stranded DNA, that are not copied yet, and they need to be protected. And that is the job of single-strand binding proteins. Okay, they're, as their name tells you, they will bind single-strand DNA and they will protect it from nucleases, right? Often bacteria have enzymes that recognize single-stranded DNA or DNA that doesn't belong to them, and they chop them up and destroy them because they consider them viral DNA, or they consider them exogenous, something that's that doesn't belong and could potentially be a threat, so they degrade it right away. So single-stranded binding proteins kind of help to protect that. So that's, just those three things. Now, in terms of initiation, which I guess we kind of didn't talk about with the leading strand synthesis, how does this process of actually synthesizing the DNA begin? So we know how DNA is melted, the template DNA is melted, how the nitrogenous bases are exposed, but we didn't talk about how does the DNA polymerase really begin synthesis. So one of the things about DNA polymerase is that they cannot kickstart the process, right? They need to have this process already started by someone else, and they can come in and then they can continue to add additional nitrogenous bases. So initially, when the first two nucleotides are added, DNA polymerase 3 can't really do that step. That step has to be carried out by an enzyme called a DNA primase. Okay, it's DNA primase, but it's actually an RNA polymerase in that it has the ability to link together two RNA nucleotides and then continue to stretch them until you have about 10 nucleotide long RNA primer. And that primer is required for further DNA synthesis. So on the leading strand, you're going to have a single RNA primer, right, added by the DNA primase. and it will then serve as that, 3' hydroxyl that the DNA polymerase needs to add additional DNA nucleotides. Now, on the opposite strand that we'll refer to as lagging strand, because it's slower, its synthesis is going to be slower than what you see in the leading strand synthesis. On the lagging strand, what we notice is that the synthesis of DNA is in the opposite direction of the replication fork. So the red arrow is showing you the direction of the replication fork, which way the DNA is being melted, which way um, the replication machinery is moving. And so because of the anti-parallel nature of DNA, only one of the two strands can grow in the same direction as the replication fork. The other strand is going to have to grow in the opposite direction. Remember, the two are anti-parallel. So the DNA strand that is growing in the direction opposite to that of the replication fork is going to grow slower, and we refer to it as the lagging strand synthesis. Now, with the lagging strand synthesis, new stretches of DNA, single strand DNA, become available, and they're not actively undergoing synthesis. So every 1,000 base pairs or so, the DNA primase is going to have to lay down a completely new primer. So lagging strand would be synthesized in multiple pieces of about 1,000 base pairs, okay? And so for every 1,000 base pairs, the DNA primase is going to have to lay down a whole new primer, and that is what's represented by these little red pieces. So lagging strand is going to have lots more RNA primers. Leading strand is only going to have a single RNA primer. Notice the topoisomerase, which I didn't, I guess, mention yet, but I did talk about it already, that you're going to have these topoisomerases, in this case, the gyrase, that is going to help release some of that extra tension that you have from unraveling of the DNA in the region of the replication fork. Okay, so you have the DNA gyrase, you have the helicase, you have the DNA primase, single strand binding proteins. You should know the function of all of them. Any questions with any of these so far?
Yeah, I have a question, Professor.
Yes.
For the lagging strand, so the DNA RNA primase also initiating the first step, correct?
Yes, exactly. But
Okay.
it's going to do it multiple times every 1,000 base pairs or so. Okay, so this is going to continue to happen, and that is what the panel C is showing you, that you have the laying down of a third primer, okay, which is going to now kickstart a new stretch of DNA on the lagging strand. So each one of these pieces is referred to as the Okazaki fragments. They're named after the individual who discovered them. And so you're going to have the DNA strands, both that are going to be copied fully, but leading strand is going to be copied as one long stretch, and lagging strand is going to be copied as multiple pieces of Okazaki fragments. So again, this is the replication process, a nice video that I like. Please watch it just to kind of see it as a process. Now, the previous image I showed you, it kind of shows you, just sort of the accessory protein, but without really overlaying what's going on with the DNA polymerase 3. So here is a more complex sort of image, also superimposing the action of DNA polymerase 3. So here you have the template strand that is undergoing obviously replication. Here is the replication, fork. The replication fork is moving towards the right of the screen. And I know that because the leading strand is growing in that direction. All right, so you have the bottom strand that is the leading strand in this case, And the top strand, that is the lagging strand. And I can also tell right away because I see a bunch of single strand binding proteins that are protecting the DNA that hasn't been fully copied yet. I see multiple primers, right, on that lagging strand. And I also see that 2 out of 3 core enzymes that were part of that sort of complex with 3 enzymes in it, notice 2 of them are actively synthesizing 2 Okazaki fragments. So, once they have completed synthesis of an Okazaki fragment because they've run into a primer that was laid down for an Okazaki fragment prior to this, they will fall off and kind of, twist and come back and latch onto a new primer that will be laid down by this primase and then continue to grow a whole new Okazaki fragment. So it's sort of this gymnastics that they're playing, especially the 2 polymerases that you see on the lagging strand. On the leading strand, you're going to have this one polymerase molecule that is going to start the process and it's going to finish the process. The other 2 are going to take turns synthesizing, alternate Okazaki fragments on the lagging strand, but all of the synthesis will occur all at once. The primase will remain attached to this complex because you're going to require its services every 1,000 base pairs or so on the lagging strand. The helicase is going to remain here because it's going to continue to break the hydrogen bonds. And then you're going to have the topoisomerase or gyrase ahead of it that's going to release the extra attention. Okay, so you're gonna have this happen on both replication forks. This single replication complex is going to copy half of the chromosome. The other replisome is going to copy the other half of the chromosome, and the 2 are going to meet in the middle and then eventually complete the full synthesis of the chromosome. Okay. Now, one of the things you have to keep in mind is that the RNA primer is not DNA. It needs to be removed. And the other thing is that these Okazaki fragments aren't exactly linked. There is a little gap between them that we refer to as a nick. Okay, so that nick needs to be sort of sealed. You need to link together neighboring Okazaki fragments after you have replaced this primer. All right. Now, DNA polymerase 3 has 5' to 3' DNA synthesis activity. It also has 3' to 5' exonuclease activity, but it doesn't have the ability to come in and digest these primers and replace them with DNA. So you have a whole new DNA polymerase that has that job. You're going to have DNA polymerase 1 that comes in, so DNA polymerase 1 has the usual activities of the polymerase 3, which is 3' to 5' synthesis and 5' to 3' proofreading. But it also has 3' to 5' exonuclease activity. What does that mean? It means that it can come at the end of the 5' end of a DNA molecule, I guess where the primer is, right? And it's going to chew out this green region, which is here used to show the primer region. So it's going to sort of latch on at the end. This is why it's an exonuclease, because it can start at one end of that DNA molecule, continue to cut these nucleotides one at a time until it sort of, frees this area. And as it's doing it, it's also sort of adding additional nucleotides. So it's not going to completely just remove the entire area and then come back and fill the gap. It's going to start cutting them one at a time and start to also grow the previous Okazaki fragment using its 3' hydroxyl until it has fully replaced all of those RNA nucleotides from the primer. Okay. However, it is unable to link together the neighboring Okazaki fragments. So that little gap remains between them, and you're going to have a different enzyme come in, use the energy from ATP to generate a bond between the 5' phosphate and the 3' hydroxyl. So this enzyme is going to be the DNA ligase that is eventually going to seal that gap. Any ideas why DNA polymerase 1 cannot seal this step or seal this nick? Any ideas? No ideas? Anything?
Is it because it needs topomerase to work with?
It needs what?
A topomerase?
No, not topoisomerase.
Uh,
No, remember it's also synthesizing. It's replacing these RNA nucleotides by DNA nucleotides. So it does have the synthesis ability as well. Remember, polymerase 1 is able to do more than polymerase 3. It's able to synthesize 5' to 3', and it does two types of exonuclease. One is 3' to 5' proofreading, and the other is 5' to 3'. So it does more than what DNA polymerase 3 can do. Let's go back to this slide, okay? The DNA polymerase 1 just cleaved off all the RNA primers, and when it removed them, what you have remaining is just this monophosphate, because the pyrophosphate has already been, released during the polymerization step, right? So the nucleotides that are part of the polymer are only going to have a monophosphate, right? And you need a triphosphate in order to provide the energy for this phosphodiester bond formation reaction. So when the DNA polymerase 1 chops off and removes, the existing nucleotides until it reaches a DNA nucleotide, the nucleotide at the end just has a monophosphate. I don't know if this image is showing you that, but it's just going to have a single phosphate group, which is insufficient for this reaction to occur. And that's why you need additional ATP and energy in order to link the two together. Yes, it will add to the OH, which you do have from the neighboring nucleotide, but it's the two phosphates that have already been hydrolyzed prior to this step as part of, this step where it was part of the polymer. All you had was a monophosphate. Now you hydrolyze it, you're just going to have a monophosphate remaining, which doesn't provide the energy needed to make the polymer. Okay. Okay. All right. So what's next? All right. So I guess this DNA ligase step is exactly showing you that you're trying to make a phosphodiester bond between a 5' nucleotide that has a single phosphate, and you're trying to link it to a hydroxyl group of the adjacent nucleotide. But because you don't have those additional phosphate groups to hydrolyze to provide the energy for this reaction that normally would for pole 1 and pole 3, you're going to need additional energy, which can either come from NAD or from ATP and this enzyme DNA ligase. So you mutate the ligase, you can't repair these nicks, they're going to remain there, right? So you're going to have this daughter strand that is simply going to be in bits and pieces. And when it tries to serve as a template, it won't really work. So ligase, again, is going to be this essential enzyme similar to DNA polymerase 3, which is going to be, required for replication to complete. Question. Yes, it Deirdre, it's because it just does not provide the energy needed to carry out that step. Okay, so next step. So proofreading, which I've been mentioning over and over again, that DNA polymerase 1 as well as 3 have this proofreading activity, which basically is the ability to step back, okay, as new daughter strand is being synthesized against a template strand. Notice here you have an A against C, so that's a mistake that was uh, just carried out. And the DNA polymerase 3 has the ability to slide backwards, which means it will go from 3' end to 5' end, cleave out this A that was incorrectly inserted, and then replace that with the G using its 5' to 3' polymerase activity. Okay, so keep that in mind that DNA polymerase 3 just has this 3' to 5' exonuclease activity. Pol 1 also has 5' to 3'. What does that mean? It means that it could essentially just start sort of, you know, cleaving these nucleotides one at a time, starting at the 5' end. DNA polymerase 3 does not have that ability. And as I mentioned earlier, this proofreading activity resides within the epsilon component of the DNA polymerase 3. And if you mutate it, you're not going to have that proofreading activity, and a lot of mutations would occur within the resulting daughter strand. Now, the final step of replication in E. coli is basically releasing the replicated chromosomes. And so why is that a problem? Because sometimes when the replication forks fall off, you can have the two chromosomes that are obviously ring shaped that are sort of linked together, okay, as sort of not simply intertwined, but, you know, kind of like links in a chain. They're just sort of sealed together, and you need to at least cut one of those replicated chromosomes and allow the other chromosome to kind of pass through that cut in order for you to resolve these chromosomes. So these chromosomes are referred to as the catenines or catenated chromosomes, and there are enzymes that specifically allow the controlled cleaving of DNA at certain spots, and then allowing the resolution of the structure. So generally it's done by a topoisomerase, because remember, topoisomerases are the ones that have that ability. They can either make a single stranded cut or they can make a double stranded cut. In both ways, they can still have you release all of that extra tension. In this case, we're using their ability to separate two chromosomes that have become linked. There can also be times where you can have these chromosomes act as dimers, where what you're seeing is the blue chromosome, which could be the parental, for example, and the red, that is the, say, newly synthesized chromosome. Obviously, it doesn't work like that. It's the semi-conservative replication. You have, both that have parental DNA as well as newly synthesized DNA. But just to kind of make you understand why it's diploid, that you have twice the genetic material, you have the two termination sites, and within the termination sites, there's going to be a recombination event that's going to occur. And there are enzymes specifically that bacteria have, for example, the XRCD recombinase, that is going to recognize the two termination sequences and allow the, again, the resolution of these two chromosomes into individual chromosomes, so that now they can be passed on to the daughter cells. If either one of these remain unresolved, then the parent cell can have twice the genetic material, but they don't have the ability to pass it on to their daughter cells. So unless they have another mechanism of resolving these structures, mutations to these enzymes will prevent these bacteria from dividing, because they don't have genetic material to pass on to the daughter cells. They have twice the genetic material, but it resides within one cell. And so it makes no sense for the cell to commit to cell division, because one of the cells is not going to receive any genetic material. Okay, so keep that in mind. So both are required. Both types of these enzymes are required, obviously, because cells can have either catenated chromosomes or dimerized chromosomes. One of the problems in cells that have linear pieces of DNA is the end replication problem. Because of this 5' to 3' synthesis activity of DNA polymerase, every time DNA is synthesized, a portion of the chromosome might be lost. So if we start looking at this linear piece of DNA and we consider the top strands to be the leading strand and bottom strand to be lagging strand, right? The top strand will continue synthesis all the way down to the very last nucleotide. On the lagging strand, however, you need to synthesize a primer every 1,000 base pairs or so. So there will come times when you would have sort of primer at the very end. And when DNA polymerases come in and ligases come in and replace those primers, they are unable to replace this terminal primer with the DNA nucleotides. And the result is that you have one of these chromosomes that have shortened in the process. And this process continues over time, and you're going to have shorter and shorter and shorter chromosomes. And so this puts a limit on how many times the cell can divide, and it's referred to as a Hayflick's limit within eukaryotic cells. So they will divide about 40 to 50 times, and then they stop dividing because they've lost so much of their telomeres. And this is why telomeres are so important, because telomeres are just a bunch of repetitive sequence that you can lose a little bit of. As long as you don't run into the coding region or into the genes, you can lose that and it's okay. But after 40 or 50 cell divisions, you have lost enough of the telomeres that if you continue to divide further, you will start chewing up some of the terminally located DNA, which could run into actual genes. And so, cells would stop dividing at that point. So how do bacteria that have linear chromosomes take care of it? So there are some bacteria, spirochetes specifically, Borrelia burgdorferi causes Lyme disease. They have these linear chromosomes, and instead of having these long telomeres that are sort of repetitive, the way they take care of this end replication problem is that they have repeats at their ends, which they can use to then sort of ligate their ends and convert their linear chromosome into a circular chromosome, right? So they're not showing you the linear chromosome without this terminal bubble that they're referring to as the hairpin loop or the hairpin end. But these are not always in a loop, as you can see it here. Okay, so anytime you have these bacteria that could potentially run into the end replication problem, we know from the example of Borrelia burgdorferi that they have the ability or enzymes that allow them to link together the 5' and 3' ends of the anti-parallel strands and convert it into a hairpin loop. And notice there's no sort of base pairing here within this sort of region. And so that it's not complementary in the same way as you had seen, or hopefully you remember from the telomeres. So in any event, the idea is that eventually this linear chromosome would also become circular, and it's copied the same way than a chromosome that is circular is copied. But the one of the differences is that you don't have the origin, you have actually two origins of replication in this case that you were seeing, because the replication has already begun here. But we saw that in E. coli, the replication fork was bidirectional. So you had two replication forks here. You had two replisomes. One was going in one direction towards the right, the other was going towards the left. And so you had bidirectional replication going on. With these chromosomes, linear chromosomes, we're not seeing that bidirectional replication fork. There's only leading strand synthesis at each replication fork, and it's going to go all the way around and synthesize the whole chromosome. Okay. All right. Questions on any of the replication material before we move on to transcription? I think we're going to leave some of this for next week. We'll see where we stop. But are there any questions with the replication so far? Okay, all right, so let's talk about transcription then. In order to understand transcription, you need to first understand the structure of a gene. Okay, so we know that using the example of E. coli, there are millions of base pairs of chromosomal DNA, and then there are about 4,300 genes on it. So how does the cell know where the gene begins and what exactly is a gene? So gene is basically just the sequence that codes for a single RNA molecule, all right? And that RNA could be messenger RNA so that it can code for proteins, or it could be tRNA, which we're going to see the role of in a moment. It's also referred to as a transfer RNA, and rRNA, which is referred to as a ribosomal RNA. So genes don't always code for proteins. There are some genes that code for proteins, and those genes are going to be messenger RNA coding genes, right? So messenger RNA is further processed and that it's translated into the protein. The tRNA and ribosomal RNA are going to function as RNA, right? And together, all three will be required for the translation process. mRNA is going to provide the code for the proteins. Ribosomal RNA is responsible for the linking together of amino acids, while reading the code from the mRNA, and tRNA is responsible for recruiting the correct amino acids based on the mRNA code. Okay, so gene is just going to be some, I'd say it's a unit of genetic information. It's a sequence of bases that are going to have a defined beginning and a defined end. So we're going to look at what that defined beginning and end is. In terms of RNA, we just saw that there are three examples of it. The messenger RNA is going to carry the message for synthesis of proteins. All three of them are single-stranded, although you can obviously see that there is some intramolecular base pairing between tRNA. Similarly, you can see intramolecular base pairing between ribosomal RNA, so that it allows you to make the clover leaf-shaped tRNA and the globular ribosome that we see. Messenger RNA generally tends to be linear, but it can also have intramolecular base pairing in what we'll refer to as hairpin loop structures. And they can sometimes promote transcription, sometimes they inhibit transcription, sometimes they promote translation, sometimes they inhibit translation. So we're going to look at examples of each as we go through it, okay? And so let's look at the structure of a protein coding gene. So the gene that will code for an mRNA. So you start off with DNA as the genetic material, which will be copied into a message or mRNA, and then that message will further be translated by your ribosomes into the language of proteins. So how does the transcriptional machinery, which is responsible for transcribing the DNA into RNA, recognize where to begin, right? Where does the transcription machinery bind on the DNA, right? We know that for replication machinery, the replisome bound at the origin of replication. That was kind of the assembly site. That is where everything began. That is where the primase is going to initially set down the first set of primers, right? And then those will be extended by the DNA polymerase. Now, in this case, where do we know that the RNA polymerase would recognize the beginning of a gene? So fortunately, you know, over the last century, people have studied the structures of genes, and we know exactly where the beginning and an end is, because we have these consensus sequences that are conserved from one gene to the another, and you make changes to those structures, and they, you know, sort of give you an understanding of what the function of those sequences might be. So when we look at the DNA, there would be regions on the DNA that will define the beginning of a gene and an end of a gene. So at the beginning of a gene, you have promoters, and at the end of the gene, you have the terminators. Promoter is where DNA polymerase would begin to sort of assemble. Okay, the machinery would begin to assemble at the promoter site. This is where the DNA strands will be melted into two individual strands, and then one of them would be used as a template. Okay, only one would be used as a template. And you can tell which one served as a template by looking at the sequence of your RNA that is synthesized using that region of the DNA as a template. So when we look at the promoter region, within the promoter region, we have certain sequences that happen to be conserved that recognize DNA polymerase binding. Okay, so already you know that if you mutate these sites, you change them significantly, they're not going to allow DNA polymerase to recognize this as the beginning of the gene, and DNA polymerase will ignore this gene. All right, so this might be a completely functional gene, if recognized and transcribed, could result in synthesis of a completely functional protein. You know, if that promoter is somehow mutated, it's not going to function. Okay, it's not going to be recognized as a gene. That sequence can stay there for all eternity. If RNA polymerase doesn't recognize it, it's not going to be transcribed. And if there's no transcript for it, there's no translation for it, and therefore no corresponding protein. Okay, so promoter has these recognition sites for RNA polymerase binding. Now, just downstream to the promoter site, there is a region that we refer to as +1. So this is the region where the RNA synthesis would begin. And often it happens to be either the A or the G, okay, base. And so A or G would be the first base that is added to the resulting RNA molecule. Okay, so +1 means that this is the first base that will be transcribed, will be copied into mRNA. And -10 and -35 are what they basically mean is that if you started counting backwards, there's no zero. You would have +1, -1, and you continue to count backwards to -10 site, you will run into this RNA polymerase binding site that we refer to as the Pribnow box, okay? Then you continue to count backwards to -35, and you will recognize the second RNA polymerase binding sites, and both are required for correct positioning of the RNA polymerase onto the promoter. And then once it's sit there, then it begins to melt the two strands and then begins synthesis of RNA starting with the +1 site. It will continue to copy the genetic material into RNA until it runs into the terminator, and at that point it will fall off. Okay, so if you look at the corresponding mRNA, what we notice is that the region corresponding to the promoter is missing. Okay, so promoter is not transcribed, it's not copied into mRNA. Similarly, terminator is missing, it's not transcribed, and it's not part of the mRNA. So promoter is serving as a site for RNA polymerase to latch on and assemble and kickstart that process at +1 site. And the terminator is where it's falling off. Okay, it's not really copying it anymore. It's just getting ready to end the transcription. You don't want to continue transcribing beyond this point. If there are mutations here and RNA polymerase continues to go beyond this, then it will continue to add more RNA nucleotides until it runs into a different terminator for maybe an adjacent gene. Okay, so that's how important both of these sequences are. Now, in addition to promoters and terminators, you're also seeing these sequences referred to as the leader sequence and the trailer sequence. So what is the role of those leader and trailer sequences? So leader sequences and trailer sequences are important for translation purpose. Just like the replication machinery and transcription machinery had start and stop sites, similarly, the translation machinery needs to have a start and stop site as well. So within the leader sequence, there is this sequence of bases that we refer to as the Shine-Dalgarno sequence. This is the recognition site for your ribosome. If ribosome doesn't find Shine-Dalgarno sequence to be part of an mRNA, it completely disregards it. It doesn't recognize it. It doesn't translate it. Similarly, you could take any random piece of RNA and you can throw in Shine-Dalgarno sequence, right, artificially, and suddenly your translational machinery, the ribosome, begins to recognize it and translate it into a protein, which might be completely nonsense, not really needed by the cell, but it's suddenly being recognized for this process of protein synthesis. So leader has that, sequence has that similar role in translation as promoter has in transcription. And trailer, again, is the region where the translational machinery or ribosome will sort of slide off. This is the end of the protein, right? So you have then the start site for protein synthesis, just like we had the first base that is added for the RNA to be G or A. We're going to have the first amino acid that is added in response to this mRNA going to happen at the initiation codon, which is AUG. You mutate that, you make, trouble for this particular mRNA, right? So ribosome will assemble here, will continue to look for sort of that AUG. If it doesn't find it, it might fall off. Maybe it will make a mistake and it will stay latched on and it will just insert the wrong amino acid or whatever, but certainly it would be a problem, okay? It may be extreme so that no translation occurs, or it might be slightly problematic because it prevents the ribosome from recognizing what the first amino acid might be, or it might be so problematic that it prevents the translational machinery from really realizing, how to read this mRNA. What's the proper reading frame? Where do I start reading it from? Okay, so they're all very, very important. So just like you have the start codon, we're also going to have a stop codon that's going to be indicative of the end of the protein, and no more amino acids are added beyond it. So all of these regions, you should know them. They are all important no matter what course you're taking. You need to understand them. They're quite similar, and we're often using bacteria and bacterial sequences to study these processes, even though they might be named differently in, say, eukaryotic cells and different organisms, but the idea is the same. And often, like I said, we're using bacteria to study these processes, so these will come up often. Now, in terms of tRNA and ribosomal RNA genes, they are going to have those same sequences. They're going to have a promoter, they're going to have a terminator. There's no need for a leader sequence, right? Because they don't need to have a Shine-Dalgarno sequence. They don't need to they aren't going to be translated into protein, so there's no Shine-Dalgarno sequence. Okay, there may be some leader sequence of some sort. Okay, but in concept, if you think about it, there's no need for that. There's no Shine-Dalgarno sequence that is needed. So the overall structure is going to be the same. They're going to have promoter, they're going to have a terminator, and they're going to have all of this coding region that will be transcribed into tRNA and mRNA. Now, in terms of tRNA and tRNA and ribosomal RNA, these genes are slightly different, that they don't just code for a single RNA molecule in terms of tRNA or ribosomal RNA. We're seeing that here one gene encoded for two different tRNA molecules. Okay, so the coding regions, there's going to be two coding regions that are going to be separated by spacers. There's going to be one promoter, there's going to be one terminator, there's going to be two coding regions separated by spacers. Okay, sometimes those spacers can have ribosomal RNA in them. Okay, so you can have the 16S and the 23S that make up the large ribosomal subunit. I'm sorry, 23S and the 5S that make up the large ribosomal subunit, and the 16S that makes up the smaller ribosomal subunit. So, you know, the spacers themselves can be made useful, and it makes sense for an organism that has a smaller genome to use as much of it as possible. Bacteria do not have introns. Their messenger RNA doesn't have to be processed the same way the messenger RNA for eukaryote is processed. But, you know, just going back to this image again, there's always going to be promoter, there's going to be terminator. You can have multiple coding regions that are separated by spacers, and those spacers themselves can have sequence for ribosomal RNA within them. And so then you're going to have enzymes that are going to come in and remove these pieces, all right, so that you have the tRNA and then you have the ribosomal RNA individually present to go on to do whatever it is that they do. And because this is RNA, you're going to have ribonucleases, enzyme that cut down RNA performing these functions. Now, if we look at bacterial messenger RNA, it's actually no different from what we just saw with the tRNA and ribosomal RNA in having multiple coding region. Bacterial mRNA can also have multiple coding regions. Generally, we think of mRNAs being monocistronic, means a single cistron or single coding region. They're generally actually polycistronic, meaning they have multiple coding regions. So when we talk about operons next week, it will make a little bit more sense. But you have a single promoter, a single terminator, and you have multiple coding regions, and the spacers within these coding regions are actually going to be leader sequences, because you need the Shine-Dalgarno sequence within the leader sequence to allow ribosome to enter. Okay, so you have the start site, you have the stop site for each coding sequence. So ribosome will recognize the Shine-Dalgarno here, you know, load onto this mRNA, recognize the start codon, start synthesizing protein until it runs into the stop codon, falls off. Okay, in order for this next coding region to be synthesized into protein, the ribosome, a different ribosome, would have to assemble onto this leader sequence within this spacer, recognize the start codon, and then continue to copy the message into protein until it runs into the second stop codon. So, you know, if you have any mutations here, or you have any mutations in the start codon, right, this spacer won't work, the ribosome assembly won't happen, and you won't have synthesis of the protein corresponding to the second coding region. Similarly, if you have maybe mutations in the stop codon, so that the ribosome doesn't recognize this as the end of the protein, it can continue to, you know, translate beyond it until it runs into a whole new, you know, stop codon, which could just be this one, for example, or it might run into one within the spacer region if it's lucky. So all of these sequences are extremely important for proper expression of genes. Now, in terms of bacterial RNA polymerase, it has multiple subunits the same way as we saw within DNA polymerase. You know, it had all of these units, alpha and epsilon and theta and tau and beta and so on. So similarly, RNA polymerase are complex as well, and there are multiple chains, and RNA polymerases are conserved, meaning there is a lot of similarity between prokaryotic RNA polymerase and bacterial RNA polymerases. So you'd remember when we talked about antibiotics, we said that there was so much similarity that we don't really have any or a large class of antibiotics that targets bacterial RNA polymerases so that we could prevent transcription from happening and kill bacteria that way. There is so much um, conservation between structures that if you try to control bacterial RNA polymerase, there will be cross reactivity to eukaryotic RNA polymerases, and it will affect our cells very similarly. So very, very conserved. Now, bacterial RNA polymerases require somebody to recognize the promoter before they begin to assemble at the promoter and kickstart the transcription process. So it's similar to what you saw with the replication, that you had the DNA A that had that responsibility of recognizing the origin of replication, and then DNA polymerase, first helicase binds, and then primase and polymerase and all of that comes in. So similarly here, you have all of these various subunits that that together make up the RNA polymerase, but then you have all of these different sigma factors that are shown here. So there's sigma 4, sigma 3, sigma 2. They don't all bind at once. They're just showing you the various sites that they interact the core RNA polymerase enzyme with. Okay, so there are different places where they'll interact with it. Now, the sigma factor's job is to come in, recognize that promoter, allowing RNA polymerase to then situate itself along that -10 and -35 sequence. And then it will actually help to melt the DNA at the -10 site at the Pribnow box. So that is where the melting would begin. And 10 base pairs downstream, remember this is -10 sequence, so 10 base pairs downstream is the +1 site. So 10 base pairs downstreams from that, the RNA polymerase would begin synthesizing the mRNA. So notice in this case here, there's no additional helicase needed. The sigma factor is helping RNA polymerase achieve that activity, right? So holoenzyme is going to have that activity. And then it also doesn't require any primer to start this process. It's able to initiate this process by itself and then continue to synthesize RNA in 5' to 3' direction, okay? It has the ability to form this DNA RNA hybrid that our DNA polymerase doesn't. It only does DNA hybrid, right? No, I'm sorry, DNA synthesis. And so once this process has begun and RNA synthesis is already underway, there's no need for the sigma factor. It can leave and it can be recycled elsewhere. And once the RNA is fully synthesized, the polymerase would be released, and it can be recycled as well, just as, of course, the DNA template is recycled. So this transcription initiation using sigma factors is an important way of controlling which genes are transcribed at what moment. So when you look at these sequences, you have the -10 sequence that has a lot of A's and T's, and it makes sense because you're going to melt that. That is where sigma factor is going to help DNA polymerase melt and begin to separate the sense strand and the template strand, and use the template strand to synthesize new mRNA, but the synthesis is going to begin at the +1 site. Now, when we compare these sequences of -10 to -35, we notice that there are different sequences present, in promoters of different genes. And there are different genes that have the, same sequences. And so what we learned is that there are different sigma factors that corresponds to different promoters. So the normal housekeeping genes, which are on all the time within metabolically active cells, are transcribed by binding of sigma factor 70. So this is your housekeeping sigma factor. And so here's the consensus -35 and -10 sequences that are given. So any gene whose promoters have those sequences will bind sigma 70. Okay, so that means not all 4,300 genes in E. coli are going to have that particular sequence. Okay, then there are other genes whose promoters have these unique sequences, and they bind a different sigma factor called sigma factor 54. And so all of these genes have been shown to be transcribed and translated when cell is undergoing nitrogen metabolism. Similarly, you have other sigma factors that recognize promoters of genes that might be involved in sporulation or in chemotaxis and motility. So this is a great way of cells to control what genes to transcribe and also kind of group them so that you're only transcribing the genes that are really, whose products are really needed at the time. Okay, so sigma factor expression is controlled tightly by bacteria. Some are kept generally at the old pole of the cell, kind of hidden away until they're needed. Once they're needed, they're brought in or they're anchored to the membrane away from the DNA, from the nucleoid region. And once they're needed, they're brought in near the nucleoid, and then they kickstart the process that results in synthesis of proteins from genes, whose products are needed to meet the current needs. So transcription initiation, I already described to you that you're going to need the sigma factor, and it's going to help the RNA polymerase sit at the -35, -10 sequence. -10 is where the melting would occur, and +1 is where the first nucleotide would be added. And then, once you have added the first nucleotide, then it's really a matter of just continuing to add RNA nucleotides from 5' to 3' direction. Now, transcription termination is going to occur in the terminator. And is there a sequence that corresponds to termination that lets the RNA polymerase know that it needs to stop transcribing? So we have looked at various terminators, and scientists have found two different types of terminators. There are terminators that are sequence dependent, so that you see a bunch of A's in the template that correspond to a bunch of U's in the RNA. Anytime you have repetitive nucleotides that are being added in the RNA, generally the polymerase slows down. So although it's able to add hundreds of nucleotides per minute, it's going to actually slow down to ensure that it's adding the accurate number of nucleotides. So if it was supposed to add 10, it adds 10, it doesn't add 9 or 11, right? So it slows down to kind of ensure the accuracy of the message that is being synthesized. And it turns out that this pause and this A-rich region in the template and U-rich region within the RNA allows formation of this stem loop structure, which happens because of inverted repeats, which I'll show you on the upcoming slides in a minute. And this somehow destabilizes this RNA DNA hybrid. And so that the hydrogen bonds break, the RNA is released, and RNA polymerase is no longer synthesizing anything. It also falls off, and DNA just goes back to being DNA. You can also have a different type of termination occur, which is factor dependent. The factor that is responsible for terminating transcription is referred to as the Rho factor, okay, Greek letter Rho. And it recognizes a sequence within RNA. So it's referred to as the Rho utilization sequence. And so once Rho factor recognizes it, it binds to it. It's actually a helicase. It uses ATP to continue to kind of run along the newly synthesized mRNA until it reaches this little bubble where transcription is going on. It uses the energy of ATP to break the hydrogen bonds between DNA and RNA, again, releases the RNA. Polymerase is no longer synthesizing anything, so it falls off and DNA goes back to being DNA. So of course, Rho protein is important for termination and this particular type of genes, and so is the sequence. If you mutate the sequence and the Rho factor can't bind, of course, that would interfere with the termination process as well. So I'll use this as the last slide. When you see inverted repeats, and you're going to see this often, it's essentially referring to sequences that are slightly away from each other, okay, that can come back, okay, and it can result in formation of these looped structures. Okay, so here you're seeing a double-stranded DNA that has these inverted repeats, which can either base pair with each other, but if these hydrogen bonds were broken and they were allowed to regenerate, these loops in other ways, you don't have to have this linear sort of arrangement as you're seeing in the original DNA strand. You could have other, arrangements that result as well. So similarly, RNA can have these inverted repeats as well. And when we talk about this hairpin loop, essentially that's what we're talking about, that RNA is synthesized. So if you can ignore the bottom strand altogether and just consider the top strand, let's say, to be mRNA, once these two inverted repeats are transcribed, they can fold back on themselves and, base pair with each other, form this hairpin loop-like structure, which can also be a signal for the end of transcription. So you're going to come across these a lot. So make sure you understand them. I'm going to stop at this slide. I know there was a question. Spacer region, Teresa. So if you're talking about the mRNA, yes, you could assume that the spacer region of messenger RNA has the Shine-Dalgarno sequence, but not the ribosomal RNA or tRNA coding genes, because they don't need the Shine-Dalgarno sequence. They are not translated into proteins. Okay. Any other questions?
Recording stopped.
No more questions? We're good? All right, guys, thank you so much for staying back. I'll post this and we'll continue from here next week. I'll see you guys next Tuesday. Take care, everyone.